Thank you so much for the introduction, and thank you all for your interest in my research. As Ryan said, I am Hannah Diaz. I'm a student at The Ohio State University in Dr. Megan Meuti's lab, and today I will be talking about seasonal changes in the abundance and reproductive activity of Anopheles mosquitoes in central Ohio. So in central Ohio, we have three major Anopheles species: Anopheles perplexans, Anopheles punctipennus, and Anopheles quadrimaculatus. We can morphologically distinguish these species by their beautiful patterns in their wing scales. While Anopheles punctipennus and Anopheles quadrimaculatus are found throughout Ohio, there's still a large area of Ohio that we find Anopheles perplexans, including central Ohio. Notably, these species are found throughout the U.S. Anopheles punctipennus and Anopheles quadrimaculatus have a range that covers most of the eastern and midwestern United States. And while a little more sporadically, Anopheles perplexans is found in many states in the eastern and southeastern U.S. And this is important because Anopheles can transmit pathogens, including West Nile virus, Mayaro virus, Eastern equine encephalitis, and most notably malaria. While there's about 2,000 cases of malaria confirmed in the US every year, most of these are usually associated with travel. However, as most of you may know at this point, there were locally acquired cases confirmed in the US earlier this year in 3 states: Texas, Florida, and Maryland. And this demonstrates that malaria could reemerge, and in part because Anopheles punctipennus and Anopheles quadrimaculatus are known malaria vectors and are found throughout the United States. So how can we predict when Anopheles might be transmitting diseases? We can predict this by observing diapause, or their overwintering dormancy. In areas that have temperate climates, decreased day lengths and decreased temperatures initiate diapause in adult Anopheles mosquitoes, females. And this allows females to survive the harsh winter temperatures and climate. Importantly, diapause is defined by a period of reproductive arrest, which then results in reduced abundance and reduced blood feeding behavior. And because of this reduced abundance and reduced blood feeding, there is reduced risk of disease transmission to humans. However, we currently do not know when Anopheles are initiating and terminating reproductive dormancy in Ohio. So how can we even tell whether Anopheles are reproductively dormant? So we can dissect the ovaries from a female, which is shown in the bottom here. The red circles are outlining the ovaries that have been dissected from a female Anopheles. And in Culex mosquitoes, reduced egg follicle length is indicative of diapause, where small egg follicle lengths indicate that a female is in diapause or reproductively dormant, and large egg follicle lengths indicate that a female is not in diapause or is reproductively active. However, we cannot use egg follicle length to distinguish between diapausing and non-diapausing female Anopheles. Instead, we can observe changes in parity or whether or not a female has previously laid eggs. So when we dissect the female Anopheles, we could find a few things. The first being that the female is nulliparous, or that she has not previously laid eggs. And we can tell that a female is nulliparous by observing the ovarian tracheoles, which are part of the mosquito's respiratory system that provides oxygen to each egg follicle. And the ovarian tracheoles, hopefully you can see, they're the very brightly highlighted, almost yellow, veiny looking structures in the top photo, and then underneath they're enlarged. And so I like to think about this as if you had a bunch of pieces of thread or fibers, and you twisted them like very, very, very tightly, almost into a strand of yarn, and then you kept twisting that strand of yarn until it was like twisting on top of itself and almost into knots, and that's what a nulliparous female's ovarian tracheoles look like. And you can kind of see the knots in the lower image there. The other option is that we dissect the female and she is parous, or she has previously laid eggs. And when females are parous, their ovarian tracheoles look more like a web or net structure. So if that piece of yarn that you just twisted up so tightly onto itself, if you took it and you pulled all the fibers apart, it's kind of a visualization of what you would see in a parous female. And you might be able to see that in the top image there, and then enlarged in the photo on the bottom. Finally, we might dissect the female and find that she's gravid or carrying fully developed eggs. And you can see the image in the top, each one of the crescent-shaped structures. Each one of those are a fully developed egg, and then there's an enlarged photo on the bottom. So only nulliparous females can enter reproductive dormancy. However, also reproductively active females can be nulliparous. So we can't use parity to distinguish between individual females, but by observing changes in the proportion of a population over time, we can determine when most females are entering and exiting reproductive dormancy. So for our study, we wanted to determine when Anopheles initiate and terminate reproductive dormancy in central Ohio. So we collected adult females from the end of September in 2021 through the middle of May in 2022, and this is during the time that most surveillance and control is not occurring in Ohio. We collected females from parks and culverts, and you can see in the map on the right the areas that we were collecting these throughout Franklin County, and an image of a park, an idea of where we collected adults, and then also the culverts. From the parks, we collected females using the CDC light traps and BG sentinel traps, and then we used a mouth aspirator to collect females from the culverts. So we found over 500 females, which is crazy, being that we were collecting females throughout the winter. We found all three species, unsurprisingly, Anopheles punctopenis, Anopheles quadrimaculatus, and Anopheles perplexans. While we found quite a few of each species in the late fall and early winter, by the beginning to middle of December, we stopped collecting as many mosquitoes of each species. Okay, so to evaluate reproductive dormancy, like I mentioned before, we looked at changes in the proportion of nulliparous females over time, and just as a refresher, if this is a new term for you, only nulliparous females can enter reproductive dormancy. So this is why we were able to look at changes in the proportion of females that were nulliparous. So on the bottom, or on the x-axis here, you can see the month that the females were collected, and then the y-axis indicates the proportion of females that we collected during that month, and these are separated by the three species, Anopheles punctopenis, quadrimaculatus, and perplexans. We found that most females entered reproductive dormancy by late September in 2021, and this is because we collected a high proportion of nulliparous females during this time. And you can hopefully see this with the gray shaded box. I can't really tell how well you can see this. I can't see it very well from up here, so I'm sorry. So while most had entered reproductive dormancy by late September, we were certain that all females of all three species had entered reproductive dormancy by mid-December. And this is because we collected 100% nulliparous females of all three species. And if they were not all in reproductive dormancy, we would have expected to see anything lower than 100%. In the spring, our results indicate that most females terminated reproductive dormancy by mid-March, and we figured this by the time that 50% of females were no longer nulliparous. So you can see for all three species the drastic change or drastic decrease in the proportion of nulliparous females. So while we had our estimates of when most females were entering and exiting reproductive dormancy, we took a closer look at some individuals just to better inform our estimates and to better understand what was going on. So here we have a graph of Paris and gravid females, and as a reminder, Paris females have previously laid a batch of eggs, and gravid females are carrying fully developed eggs, and neither Paris or gravid females can enter reproductive dormancy. Hopefully you can see the gray shaded box in the graph, and that indicates our estimated period that I talked about before when females were entering and exiting reproductive dormancy. So in the fall, we collected quite a few Paris and or gravid females. We collected gravid females until the middle to end of September, but didn't collect any gravid females afterwards. We did, however, collect quite a few Paris females. However, it's unclear whether these females were actually reproducing or they had previously reproduced and were hanging out and kind of waiting to die from the cold temperatures or lack of resources. Like I said before, by the middle of December, we were no longer collecting any females that were reproductive. They were all in dormancy during this time. So this allows us to, because all females were in reproductive dormancy during that time, we know that any females that emerged after late February had undergone diapause and terminated diapause and were again reproductively active. So by the beginning of March, we collected reproductively active females through the end of our collection period. And this is kind of interesting, something I wanted to point out. We predicted that most females terminated reproductive dormancy by the middle of March. However, we were collecting Paris females as early as March 2nd. And because females were Paris at this time, they had already developed and laid a batch of eggs, indicating that some females were terminating reproductive dormancy by late February in 2022, which is crazy. Again, to take a closer look at just some individual females, we looked at blood-fed mosquitoes, and because females are blood-fed, this indicates times when females might be transmitting diseases. Again, the shaded box is our estimated time of initiation and termination of reproductive dormancy. So in the fall, we collected blood-fed mosquitoes until the end of October, which is about a month outside of the current surveillance and control season in Ohio. So this indicates a time when we aren't surveying or controlling mosquitoes, and Anopheles mosquitoes are still out and blood feeding. In the spring, we collected blood-fed mosquitoes as early as April 6th, and this was after our estimated time of that most females terminated reproductive dormancy. So this makes sense. However, if you remember from a few slides ago, we were collecting Paris females as early as March 2nd, and Paris females again had previously laid a batch of eggs. And so in order to produce that batch of eggs, all of the females that we collected during this time, from March 2nd to March 17th, all of these females had to have taken a blood meal, again indicating that some female Anopheles were host-seeking and biting by late February in 2022. Okay, so to summarize the major findings of our study, host-seeking and reproductive activity extends well beyond the surveillance and control season in Ohio. We found that most females had entered reproductive dormancy by late September. However, we were still collecting blood-fed females through the end of October, indicating a time when Anopheles mosquitoes are still biting and potentially transmitting disease. In the spring, we predicted that most females terminated reproductive dormancy by mid-March in 2022. However, there were females that were terminating reproductive dormancy, biting, and host-seeking by late February. And I think I have time. So some other fun, non-intended results of this study that I thought some of you might enjoy. Concerning identifying Anopheles perplexans. So Anopheles perplexans and Anopheles punctipenis morphologically are very similar. Currently, we distinguish these two species by the light band at the top of their wing, where Anopheles perplexans have a shorter light band of wing scales and Anopheles punctipenis have a longer light band of wing scales. However, this can kind of be fuzzy, as sometimes the Anopheles perplexans or Anopheles punctipenis aren't as short or long as the images shown here. Sometimes they're kind of in between, so it's hard to tell what species they are by doing this morphologically. So instead, we could genetically identify Anopheles perplexans so that we could distinguish from Anopheles punctipenis. Unfortunately, the genetic sequences of Anopheles perplexans have not been thoroughly examined. However, there are two important molecular diagnostics that we could use to distinguish between most Anopheles species. So the first one being the CO1 sequence, and the second being the ITS2 sequence. So a recent and previous study found that you cannot use the CO1 gene sequence to distinguish between Anopheles perplexans and Anopheles punctipenis, as the CO1 sequence in these two species are the same. In our study, we attempted to use the ITS2 sequence to distinguish between these two species. However, we found that the ITS2 sequence of Anopheles perplexans and Anopheles punctipenis was identical. So currently we don't have a great way to morphologically distinguish between adults, and the genetic differences between these two species are currently still unknown. I would like to thank my study co-authors, Dr. Megan Meuti, my advisor, Dr. Laura Pomeroy, Dr. Alden Sipperstein, our undergraduate researchers who helped in setting traps, collecting traps, identifying mosquitoes, Sydney Robert and Lucas Sarko, our research collaborators, Leanne Garrett, who provided traps and baits to collect mosquitoes, and then our funding from OSU, CFAES, NSF, NIH, and USDA. And that's it. If anybody has any questions, I can. Yes.
Why did you choose to focus on Aedes instead of Culex or another species?
Yeah, so we chose to do Anopheles instead of Culex or any other species because Anopheles and Culex are the only two genera in Ohio, or maybe anywhere, that diapause as adults. Aedes diapause or overwinter as eggs. So it's a little easier to go seek out adult mosquitoes than maybe it would be to go looking around for eggs. And my project was with Anopheles because Dr. Alden Sipperstein, he looked at Culex mosquitoes, which if any of you were at OMVCA meeting last year, you might remember his talk about some of that. But yeah, that's ultimately why I looked at Anopheles mosquitoes. They're pretty understudied just because they're not not considered like the most medically important genus or species of mosquitoes in Ohio right now. But we did find some interesting results, and coincidentally, there were, and unfortunately, locally transmitted cases in the US, so.
Was there any correlation between the first pairs of females in early part of the year, like late February, March, and the location collection site?
So we actually didn't look at the correlation between like where they were collected and when they were exiting reproductive dormancy. So in Alden's study with Culex mosquitoes, the microclimates were so there were so many things that were going into these microclimates that it would have been hard to like rule out what specifically was affecting or leading to these like mosquitoes terminating reproductive dormancy like earlier than others. So we didn't really look at that. And even if we did, we collected like so few mosquitoes in the spring because most of them had died at that point that we probably wouldn't have had enough to find anything significant. Yes.
Maybe I'm just unaware of like the life cycle, but did you ever find any like blood-fed Anopheles like resting, or do they not rest to hang out in culverts and stuff that soon after they've taken a blood meal?
Yeah. So Alden actually. Thankfully, he collected most of the mosquitoes from the culverts. I had the opportunity to go with him a couple times. It's crazy if you never crawled around in culverts and collected mosquitoes. Do it once, it's fun. Anyways, but yeah, so I know in the spring and then the early summer, he was collecting blood-fed mosquitoes from the culverts. And he saw that with Culex mosquitoes as well, that once they took their blood meal, they were going back to the culverts and kind of hanging out, hiding away from whatever, waiting to lay their eggs somewhere. So yeah, definitely during the end of the spring and early summer, and probably into summer. So.
Did you do any analysis to see what they were feeding on then, or are there plans to do any?
I haven't done any. They are saved in the freezer. So I know Alden was working on some blood-fed stuff, so I'm not sure if he's going to continue with any of the non-Culex species. I'm sure somebody will do something with them at some point. It would be interesting to look at those, though. I unfortunately did not. Thank you.